Hi, this is Miss Litton, and again, this is my wonderful Period 1 class. Say hi. Hi. All right, so this is the part 3 of Chapter 19, Taxonomy, Systematics, and Phylogeny, and we are focusing on phylogeny. So we use characters to organize organisms into their phylogenetic tree. So could I have my youngest bio buddy tell what a character is? Go ahead. Tell them right now. Go.
I'm more worried about
all right. So again, revisiting, we just had a quick review on the 3 domains that we had and what distinguished them from each other. And so now, if we look at constructing a phylogenetic tree, we look at the ancestral characteristic, and in this case, it's the ability to do what? Look at the slide. What is it? Ability to what? Climb trees. Right here. This is the ancestral character, right? The ancestral trait that we're looking. I'm going back here. Character is any structural, chromosomal, or molecular feature that distinguishes one group from another. So here we're looking to see, can they climb a tree or not? And then they diverge. One group went left, another group went right. If they go left, they climb those trees and they run along the branches. But if we go right here, they swing from limb to limb. These are derived characters. So their ancestral character is that they can climb the tree. So you look at grouping organisms based on their shared derived characteristics. Derived characteristics. We share those same derived characteristics. We will be in a group together. So now let's step back a little bit further and bring back our reindeer that we so love of all the different groups. So they have a ancestral trait that's the same. They have mammary glands. But other than that, do they have other traits that they share? Are we going to classify them right here whether or not they have fur or hair? No, we're not going to look at those traits. And as we start to put them in their phylogenetic trees, we're probably not going to classify something on a trait or distinguish it if it pops up again later and re-evolves later. It probably only evolved what? Once. The odds that a trait, that you lose it and then re-evolve that trait, probably that doesn't happen, right? We're going to go with the simplest explanation when we start to group our organisms. So as we step back again, I want to remind you how we, these phylogenetic trees, and when we looked at this before, you're definitely looking at characteristics, but we are applying where they came from, their evolutionary line. So this process is called cladistics, and this would be an example of a cladogram. So you have a shared trait here, but starting at this branching point, from here up, they all have what? Jaws. From here up, they all have jaws. It's not something that we're going to, whatever trait we pick, it's just going to evolve one time. So we have to leave off that hagfish, the jawless fish. But now we're going to dump off perch because perch doesn't have what? Lungs. Okay? And now we're going to dump off the salamander because he doesn't have what? Claws or nails. But everybody else from here on up does. We now are going to come here. Now here, this is a problem. Birds are a problem based on our current classification system. So now from here on up, we have fur and mammary glands. He doesn't. Maybe he belongs in this group right here with lizards and other reptiles. And that this is a derived trait that came later, but that's the only thing that's distinguishing him. And that is the argument, that birds shouldn't be in their own separate class. Okay, but we'll talk more about that later. But this is an example of a cladogram. And when you organize one, okay, you're looking at those shared derived characteristics, and could blue explain this simple diagram right here? What did they do in this simple diagram? Go ahead. screws in this? Okay, so it's all coming full circle. So all of them share A, that's your ancestral trait. Now they have some derived characteristics, right? This grouping over here all has one thing. They all have C. We're not going to see a split here and then have A, C, E, and now B over here. Because it's probably not going to evolve two different times. Probably they were related if they both have that B trait. So when you look at cladistics, you can make a chart. And when you make this chart, when you're trying to establish a cladogram, you look here, notochord in the embryo, all of these species had that. So we're probably, if all of them have that, we'll call that the what trait? The ancestral trait, right? Then we start to look, who has vertebrae? Okay, who has lungs? Who has a three-chambered heart? Who has internal fertilization? Now look at this one right here. We can see a pattern in these three, but these parts right here, the four bony limbs, long cylindrical body, we're not finding that in the same way. Maybe we're not going to take that into consideration. Maybe when you look at your nails, you're not going to look whether it has the pointy or the flat end. Maybe you're going to dismiss that trait, or maybe you will include it. What the top looks like, maybe you include that, maybe you don't include that when you're constructing your cladogram. Okay, and when we look here, the ancestral characteristic here is notochord, then we left off lancelet, but everybody here has vertebrae. From here up, they have lungs and a three-chambered heart, both those traits. From here up, they have amniotic eggs and internal fertilization. We didn't take into consideration the long cylindrical body, right? We didn't take that. We didn't find a clear pattern in that. Okay, oh, you already know that. Okay, so which option makes the most sense when you look at this picture? Yay! Our first Sunday. Good job. That's right. You picked the right one. I was hoping you would. I've had those slides ready, that's right, for every single one, but I haven't been able to use it yet. I'm just kidding. Okay, go to interpreting phylogeny. Constructed from traits that are shared by and unique to a taxon and their common ancestor. When a new character or trait evolves, then the tree branches. Then the tree branches. When you get a new trait, tree branches. 1 second. Ancestral is shared by all in the lineage, and you don't use that to distinguish them. Where a derived trait is new, it is not found in the common ancestor, and it's used to distinguish groups. All right, and so cladistics is a method that uses shared derived traits to develop a hypothesis of evolutionary history. A cladogram reflects the evolutionary history. A common ancestor and all of its descendant lineages is called a clade, or also called a clad, C-L-A-D-E. Slate, could you please distinguish between an outgroup and an in-group? Okay, and we use the principle of parsimony when deciding which traits to use. The minimum number of assumptions is the most logical. The minimum number of assumptions is the most logical. Go simple. Do not have a character evolving twice. The minimum number of assumptions is the most logical. Go simple. Do not have a character evolving twice. The best cladograms have the fewest and I have there in quotes, unexplained shared derived characters. The fewest things that we need to explain or go, I don't know why this is here. You have to be very careful of convergent evolution. Very careful of convergent evolution. So here we have a problem. So traditionally, birds have always had their own class. You had class Reptilia and class Aves. But here's a problem, because now we have 3 different things here, right? So what is it? All of a sudden we've branched into a trifecta. Either you have this or you don't have this. All of a sudden we have this whole new trait evolving. So strict Cladis would put all of those together, okay? Crocodiles and birds all share that common ancestor, and that's what I have here on number 5. I think it's the water in the neighbors. So using fossils in phylogeny, you're limited by the types of structures that are fossilized. You're limited by the types of structures that are fossilized. So it's difficult to know who is related. Okay, you do look at morphological traits. You look at homologous structures. You're very careful of analogous structures that are a result of convergent evolution. We've talked about that. So morphological traits use homologous structures, and be careful of analogous structures. And if possible, you're going to look at behavioral traits if it can be identified. If it can be identified. Okay, and then we've talked about this idea before about a molecular clock, and you're looking at mutations. You could be looking at mutational rate. You could be comparing sequences of DNA. You could be looking at proteins. So molecular clock, the number of neutral mutations not based on selection. You can look at amino acids and proteins. You can look at nucleotide sequences in DNA, including mitochondrial DNA. That should be rapidly, not slowly. Mutates rapidly. Sorry about that, you guys. So you can look at protein comparisons, looking at the sequences of the amino acids. And mitochondrial DNA mutates 10 times faster than nuclear DNA. Use it for closely related species. So fix slowly to rapidly. Do you see where I am? Did you fix it? Okay. Okay, and while you're finishing that, did you see your review that I have up here, the summary? Yeah. And I put in a how to read a cladogram. Do you see that? And I compared bacteria, archaea, and eukarya. See the chart? You are welcome. Yay! Another Hundy. I'm so proud of you. Okay, we are kind of at the end, okay? Now, to get us ready for our cladogram, okay, and we do have a quiz. I know that we're just going to work a little bit on it. You probably won't finish the process entirely. I want you to revisit, because it'll be easier to learn on nails and screws than actual organisms. I have a little chart for you. And the first thing you want to do is start to look at your chart. And we have time I set aside for us to finish chapter 19, which we, in essence, have. We have all the content. But on Friday, and we're going to introduce a lab on that day. as well. And I'm not going to have it due that day, that lab. Okay, your cladogram, yes, to prepare you for the lab, but you're just doing that in your group, and you can get started on that now. I'll talk to you more about that lab in a little bit, but I want you to take a look, and this will review cladograms as well on the back on how to set up a cladogram and the basics of that. And I want you to start looking at your nails and revisiting that idea of how you would maybe adjust your current diagram into a different one based on shared derived characteristics. And I want you to use the other side, and you're eventually going to take a picture of that. And if you're not ready to take a picture of it today, that's fine. We can work on it Friday. But I do want you to go into your team, and I want you to go over at least the back side of cladogram basics. Okay, questions or concerns? Okay, and then we're going to throw into a Google slide your before and after and make comparisons. And we will evaluate the best cladograms. We will see.
Oh, yeah. What do you...
All right, go to your team. Okay, make good choices. You're super smart.